This video is an approximation of the so-called diffusion noising. Denoising, which was developed for images and recently applied to the tiniest protein structures. By adding variable conditions to the denoising processes and thanks to the computational training of the programs with banks of millions of images, after the denoising and starting image can be converted in several ones randomly defined. Still in their beginnings, the diffusion noising and denoising of images is been applied to the multiple tiniest protein structures in many different ways, including health-related diseases such as antibiotic resistance of virulent bacteria, an increased worldwide health concern. In the following example, we were targeting a bacterial Regulon protein responsible for the expression of hundreds of bacterial virulent genes. Superbacteria are becoming increasingly resistant to many of the most potent antibiotics, including those used in mixtures. We can see the tridimensional structure of one Regulon. We can see the Regulon protein to the left and to the right the complementary proteins randomly generated in a few minutes by the diffusion processes drawn in different colors. Within the Regulon protein we chose to target or condition the random diffusion its top to bottom longest alpha helix. We chose that target because this part is necessary to form the dimer structures that talk to the bacterial DNA to tell it to express hundreds of virulent genes. Among the millions of possible denoised structures, the diffusion programs were capable of selecting several protein structures complementary to the bacterial Regulon alpha helix. The best one predicted an affinity binding strength of around 1.5 picomolar, an affinity low enough to block the bacterial Regulon. In vitro bacterial inhibition evidence and physiological fitting are further required to validate these structures.